Okay, so here we're going to continue to talk about regulation in eukaryotes. And so here we're going to talk about extracellular signals that can help to regulate genes as well as transcription factor regulation. So as an example of extracellular signals, we can have our steroid hormone receptors, and these are going to be monomeric type 1 receptors. And they are going to be complex with a heat shock protein 70. So we have a heat shock protein 70 complex with our hormone receptor. And then when our hormone binds, it's going to dissociate the heat shock protein 70. So heat shock protein 70 is sort of a placeholder. And then the hormone receptor, the hormone complex will form a dimer, and this dimer will go to the nucleus and bind to hormone response elements. And these hormone response elements are going to be part of the actual DNA sequence that's going to turn on transcription of genes. So some examples of these are going to be estrogen, progesterone, our androgen, and our glucocorticoids. And they're all going to use the same function. Now, another type of receptor for an external signal is our thyroid hormone receptors. These are going to be type 2, and they're always in the nucleus. They're always going to also be associated or bound to the hormone response element in the DNA. So here we have our thyroid hormone, and it's going to cross the cytoplasm, enter the nucleus, and it's going to bind to the thyroid receptor. Now, the thyroid receptor is always going to be associated with a co-repressor. And so this is going to be sort of a complex that is always bound to the DNA. And then when the thyroid hormone binds, this is going to release the co-expressor, and it's going to recruit a co-activator, and then this is going to turn on genes that your thyroid hormone wants to turn on. Now for here we have our thyroid receptor, and this is going to be bound to the retinoid X receptor. And so that is going to be, again, it's sort of a dimer. The RXR and the thyroid receptor form this dimer that is suppressed and the genes are off unless the thyroid hormone is present that can relieve that co-repressor and then turn on genes that the thyroid hormone will initiate. So hormone receptors are going to interact with the DNA via that zinc finger we talked about. And so remember, our zinc fingers are going to have zinc domains that do not interact with the DNA, and the other domains are going to interact with the DNA. So DNA binding domains are going to be made up of 2 zinc fingers. And so you're going to have this region that's going to bind to these fingers, which is going to be conserved, so the zinc fingers can bind to them. And again, our steroid receptors are all examples that will use this zinc finger to interact with the actual DNA. Now, you can also have regulation at the level of transcription factors. So we talked about transcription factors. For example, we had transcription factors activated from insulin signaling, and this led to your MAP kinase. We had it through glucagon signaling, through our CREB, our cyclic AMP binding proteins from pKa. And so these transcription factors all are going to find their way to the nucleus, and they're going to either stimulate or inhibit the production of genes. And so these transcription factors can have multiple targets, and they're going to often be coupled with a similar response. So, for example, insulin is going to turn on a different set of genes than glucagon, and glucagon is going to turn off a different set of genes than insulin would turn off. But just basically how this works is that you will have some sort of transcription activator. So this is going to turn on gene A. It's also going to turn on gene B. And gene B is going to produce protein B. Protein B will then go back to the nucleus and turn on another set of genes, gene E, F, and G shown here. And so you can have the original transcription factor turn on other transcription factors by making them through translation. And so just overall, in summary, understand that prokaryote gene expression is going to be regulated at initiation. And so you're going to have a series of genes that are going to be made up of an operon. And the operon will have a single promoter, so it's polycisteronic, can make many proteins. And they're going to have activators and suppressors. Housekeeping genes are always going to be on in a prokaryote unless they're turned off. Other genes are going to be off until they're turned on. Eukaryote, we have gene regulation at different levels than prokaryotes. So we have DNA at the structural level, how the DNA is packed, histone modifications, DNA methylations. There's also going to be regulatory sequences that can be very far from the gene that it's regulating. And you're going to have signals from the outside that are going to activate transcription factors. And these transcription factors can then go on and turn on genes.